Hi everybody, welcome back. Miss Calabrese here. Today we're getting into our second video of the series, talking about microscopy and visualizing microbes. All right, so when we're talking about microbes, we're obviously talking about structures that are really, really small. So if we look at this scale here, to the far right side of the scale are things that you can actually see with the naked eye. So for example, the egg of a frog is a pretty large cell. You wouldn't need a microscope to be able to see that. But the more we move to the left of this picture here, we're getting to smaller and smaller things. So the next kind of jump down here is a human egg, a grain of pollen. Those are obviously very small. And if we get down a little further, we can get to kind of your average animal cell or plant cell size. So those are relatively large cells. If we get even smaller, we can get into some of the smaller cell types like your red blood cells. And then when we get down to this like 1 micrometer range, so that kind of funny shaped symbol down there means micrometer. So when we get in that 1 micrometer range, that's when we start seeing bacterial cells. So bacteria are very, very small, much smaller, 10 times smaller than your regular animal cell or plant cell would be. Right? And if we keep going towards the left, if we keep getting smaller and smaller, then we start to see some viruses. So viruses can even be 10 times smaller than bacteria. So very, very small particles here. And then if we keep moving to the left, we get down to individual molecules, so a single protein, a single limit, or a single lipid, and then all the way down to a single atom at the very left-hand side of the scale, right? So certain things on here you're going to be able to see with a light microscope, and many things you will not, right? So a light microscope is pretty good for seeing cells. So we can see eukaryotic cells like the plant and the animal cell here. We can see bacterial cells with a light microscope, but anything subcellular, smaller than a cell, for example, like even the contents of the cell, the stuff inside of it, you're going to need a much fancier microscope, an electron microscope, to be able to see those subcellular structures. All right, so to understand how microscopes work, we need to know a little bit about light. So just briefly, light is made of waves of photons. So photons are tiny little packets, little particles of light. And those particles move in a wave. And there are a few terms that you should understand that we associate with those waves. So the amplitude here is the height of the wave, right? So the amplitude is how tall that wave can get. The wavelength is the actual length between peaks of the wave. So between this peak here and this peak here, that's going to be our wavelength. The more wavelengths you have per unit time, so if we're adding time into the equation, then we can talk about frequency. So frequency is when you have a lot of wavelengths in the same or in a short amount of time versus a small amount of wavelengths in that same amount of time. All right, so wavelength is what lets us know what color light we're looking at. So for example, the visible spectrum, which means all of the light that your eyes are able to perceive, that's usually roughly between 400 nanometers and about 750 nanometers wavelength. So that's the visible spectrum. And so that's just this tiny little box here. Everything to the left or right side of this box is going to be outside of your visible spectrum. So either the wavelengths are going to be much, much smaller or much, much bigger, right? So as we go this way, our wavelengths are getting bigger, so above 700. And if we go this way, our wavelengths are getting shorter, so below 400. All right, so we need light in order to visualize with a light microscope. So we pass light through a specimen, and then the light that was passed through that specimen goes through a lens, and that lens is going to bend the light towards our eye. And then that light is going to enter the eye, and then you can perceive that as a visual signal. Um, the lens not only bends the light, but also makes that object appear bigger than it is. So that's the first main component of microscopy is magnification. So magnification is just making something appear larger than it is. And most light microscopes have two lenses in them, at least. So those would be the objective lenses and the ocular lenses. In addition to magnification, we also need resolution. So resolution technically is the ability to distinguish two objects that are close together. It's basically being able to know where one object ends and the next object begins. So think of it as like the crispness or the clarity of an image. If an image looks really fuzzy, then it's got low resolution. If the image looks really sharp, then it's got high resolution. So we can't just have magnification because as we magnify, things will get blurrier and blurrier unless you're also adding resolution. So we need both magnification and resolution working together. Two main things that affect the resolution of a microscope are going to be the wavelength of the light that's passing through it and the numerical aperture of the scope. All right. So resolution is something that we can adjust up to a certain point, and then the air kind of gets in our way. So if we look at this picture down here, say we've got a slide here. So here's our slide, and you've got bacteria on the slide, and we've got a light source down here that's shining light upwards into this lens here. But as the light shines upwards through the slide, through the bacteria that are on the slide, and then upward into the lens, some of that light gets lost. So it gets kind of scattered. It's kind of going out sideways. One way to prevent that scattering of light is to add some oil. So if we add oil between the lens and the slide itself when we're using our microscope, that can help us bend some of these rays of light. So instead of escaping, they go up towards the lens itself, and that's going to improve our resolution when we're at really, really high magnification. So that's called oil immersion because we immerse the field in oil to stop the scattering of light. Right? And the last concept that we need for microscopy, in addition to magnification and resolution, is contrast. So contrast is basically the difference between an object and its background. So being able to see some difference between the color of an object and the color of the background. All right, so let's get a tour of the anatomy of a microscope here. So number one at the top here is the eyepiece. So the eyepiece is the part of the microscope that you look into. These kind of microscopes, like the one pictured here, have ocular lenses in there that are able to magnify what you're looking at by 10 times. So make it look 10 times bigger than it actually is. Number two over here is your nose piece. So the nose piece is holding multiple different objective lenses. So objective lenses are these structures that are kind of hanging down from that nose piece. And usually there are 3 or 4 different objective lenses here. The objective lenses usually have different levels of magnification. So you can have ones that magnify 4 times, 10 times, 40 times, or 100 times. So that's going to really increase your magnification power. Um, Going on numerically, let's look at the number 4 here, which is the coarse adjustment or coarse focus. So this is a large knob on the side of the microscope that basically lets you move the stage up and down. So we'll skip to number 6 and talk about the stage real quick. So this flat surface here where you place your slide is called the stage. The coarse adjustment and the fine adjustment as well, so numbers 4 and 5 here, both of those dials help move the stage up and down. Coarse adjustment is good for moving the stage up and down quickly when you're using low power magnification. Fine focus moves the stage just ever so slightly when you're using high power. All right, moving on down. Next up here is the X and Y stage control knobs. So these are knobs that kind of hang from the bottom of the stage and let you move the slide around back and forth, side to side on the stage. The rheostat is what you use to adjust the amount of light. So the light comes out of the illuminator, which means there's just a little tiny light bulb in here, and you can control the brightness of that bulb. And then right underneath here, between where the light is and where the stage is, we've got a structure called the diaphragm. I'm sorry, diaphragm and the condenser both together. So the condenser is a structure that's going to take the light that's coming from the illuminator and condense it into a single beam of light. So it's preventing some of the light from scattering, so it's condensing the light. And then on that condenser, there's a little dial that is called the diaphragm, and that diaphragm allows you to limit the amount of light that comes into the condenser. So condenser condenses the beam of light. The diaphragm controls how much light is going to be condensed. All right, so that was a light microscope. Moving on to an electron microscope, again, these are the type of microscopes that you would need to look at very small structures. So the organelles within cells, even some large macromolecules we can visualize with electron microscopes. But these are very expensive equipment, and they don't use light. Instead of using light, they shoot beams of electrons. So instead of here we can see the light microscope where we've got a light up top, and that light is going to be shining through a number of lenses. As it shines through those lenses, the lenses are going to magnify what we're looking at. Um, Similar concept here with the electron microscope, but instead of light, we're shining a beam of electrons through various lenses. And then the final result, instead of going directly through a lens into your eye, goes directly onto a screen that you can then look at. So this is good for things that are very, very, very tiny. All right, so two major types of electron microscopes, transmission electron microscopes, or TEMs, and then scanning electron microscopes, or SEMs. So transmission electron microscopes shoot that beam of electrons and they go straight through whatever it is you're looking at. So they pass directly through your subject. So you can tell areas of density by seeing darker areas because fewer electrons will have passed through those denser areas versus lighter areas that were more easy for the electrons to pass through. A scanning electron microscope, basically you're still shooting a beam of electrons, but prior to that you're going to coat your surface with a thin film of metal so that the electrons have metal to bounce off of. So this is really good for seeing kind of like a 3D exterior picture of whatever you're looking at. So this looks a little bit more 3D, lets you see the outside surfaces of the sample, but not necessarily the inside. Okay, so let's get into a few different microscope techniques. So this first one is a wet mount. So a wet mount means basically just putting a specimen on a slide with a drop of liquid. So this could be, a drop of pond water that you get from outside, and it's going to have different critters swimming around in it. Once you put that drop on your slide, then you would cover it with a cover slip and look at it under the microscope. A lot of times we use wet mounts when we are looking at things that are still alive, because we don't necessarily have to kill them to do a wet mount. And that's also one of the reasons why many times wet mounts are unstained. So unstained means that we haven't added any color to it, any artificial coloring, because a lot of times those stains are going to be toxic to whatever might be living in there. So a wet mount is good for looking at things that are still alive. If you're looking at something that is actively swimming and you want to view motility, then there's a specific type of wet mount called a hanging drop. And a hanging drop is just a wet mount on a slide that's got a little extra room in it for critters to swim around in. Okay. As far as dyes and stains go, um, so we mentioned before that along with magnification and resolution, one of the most important things about microscopy is contrast. So you need to be able to tell where an object ends and where the background begins. And that can be tricky for a lot of tiny things like bacteria because they don't have any natural color to them, right? So they're essentially clear. So if you're looking at something very, very tiny that's also clear, it's really difficult to see. So a lot of times we will add color, add a stain in order to be able to visualize those to increase the contrast so that we can see better. So in this case, in this picture here, we're adding some bacteria to a slide, letting it dry. So just usually let it air dry. And then we're going to do something called heat fixing, which means pass it through a flame. And passing it through a flame is going to help the specimen stick to the glass slide. So that's an important step. And then this last step here would be to add the stain to it, rinse it off, dry it, and then you can put it on the microscope and you'll have a well-stained specimen that'll increase your contrast, your ability to actually see the microbes on that slide. All right, so a few different types of stains here. So we can categorize stains by their pH. So they're either basic, which means they have a high pH, or they're acidic, which means they have a low pH. Basic dyes have a positive charge to them. So they're going to be attracted to the negatively charged components of the outside of a cell. Acidic dyes have a negative charge to them. They're going to be repelled by the negative outside of the cell. So a basic dye is what's going to give you an image like the image A here, where we can see that each one of these individual bacteria has been stained. It's taken up that stain and we can see the shape of the bacteria. An acidic dye is going to give you kind of like, think about it as like a photo negative. So you're coloring in the background, but you're not actually staining the specimen itself. So that would be like what you're seeing in image C here as an acidic stain. All right, a little bit more terminology here for staining. So simple stain is one that just involves one single stain. Differential stains involve two or more different colored stains, and we use them usually to distinguish different types of bacteria. So a couple common types of differential stains in this chart here are gram stains. We'll talk about what that is in a second, and acid-fast stains. So these are really good for distinguishing different types of bacteria, and they contain more than one step. So it's not just one single dye, it's multiple. All right, so gram staining is our most common staining for bacteria. This lets us put bacteria into two major groups. Either they are gram positive or gram negative. And that the staining is really a reflective of what's going on with the cell walls of these bacteria themselves. So the stain lets us in, clues us in as to what's going on with the structure of the cell wall based on which parts of the staining process adhered to the actual cells. And we'll talk in more detail about how gram staining works a little bit later. All right, acid-fast staining. This one is good for staining bacteria that don't pick up a gram stain as well. So these ones are typically a little bit harder to stain. So things like Mycobacterium, Microbacterium leprae causes leprosy. It's got this kind of waxy coat on the outside that makes it difficult to pick up stains, but an acid-fast stain will work for staining Mycobacterium leprae. All right, here's a capsule stain. So when we're talking about a capsule in this context, um, if we're looking at this picture right here, this is, I'm going to kind of outline with my highlighter here the actual bacteria here so we can see that the bacteria there, it's a diplococcus, so two of them together. Those bacteria are then surrounded by this much thicker layer of a protective coating here, and that's called a capsule. So capsules are really important structures to help bacteria adhere to surfaces and also to protect them from being eaten by white blood cells, for example. Um, so if we want to visualize that capsule, we often have to use a negative stain in order to actually see the capsule around the outside of the cells. So that's the capsular stain. All right, another type of stain here is an endospore stain. So endospores are structures that we find inside some bacteria that allow them to survive in harsh conditions. So an endospore, think of it as like almost like a little tiny egg that's on the inside of a bacterial cell. So where if we kill the bacteria with, say, a disinfectant or something, that bacteria, that living part of the bacteria will die. But that tiny little egg, that tiny little endospore can survive and just wait it out until conditions are good again, and then he can hatch out a whole new bacteria from that endospore. So endospores, as you can imagine, make it hard to get rid of certain types of bacteria. The only bacteria that have it are those in the genus Bacillus and the genus Clostridium. but that makes them really difficult to get rid of because they can form those endospores. So to visualize endospores, we use an endospore stain, which is usually a malachite green color. All right, and a flagella stain. So flagella stain, just what it sounds like. It's a stain that basically creates a coating around the flagella of an organism so that we can see those flagella a little bit better on the microscope. So flagella are typically very, very slender, very delicate structures that are hard to see under a microscope. But if we coat them, then with this flagella stain, then they're much easier to see. All right, a few questions here before we wrap up. So, what components of the microscope are used for magnification? What type of stain should I use if I'm going to stain a specimen directly? And what mounting technique would I use to view microbes that are swimming, so alive and swimming? All right, there's your answers. So the parts of the microscope that are responsible for magnification are the lenses. And a normal light microscope is going to have ocular lenses and objective lenses. What type of stain should we use to stain specimen directly? You're going to use a positive or basic stain. And then if we're looking at a microbe that's swimming and we want to be able to see it swim, then we'll use a specific type of wet mount called a hanging drop. All right, so I hope this was helpful. Let me know if you have any questions.